Scissors, mouse. Australia was good.
Yeah.
You get to go back.
Yeah.
They're not just separated.
Every semester.
You used up your little break.
Yeah. Yeah. So no, I'm really excited to do that.
Yeah, I just found something there.
Now I get it.
There's no.
Everybody's gonna be excited from life to Australia. Just one of my seniors.
Good, how you doing?
Trying to come down.
What?
Looks like it's all go.
Yeah, no, give you.
So we'll do something. Do something.
Not because I want it.
Yeah. He was originally going to fly for wherever he is. It's still cheaper to fly to wherever I am. From less. So how many classes do you teach?
2 involved.
Are they?
No, small. Oh, God. So the first year round algebra and then advanced small algebra. No, that's right over my shoulder. We'll see how that goes. But
So they're 10, 20 people?
yeah, it's yeah. I'm giving the undergrad 12 for
Starting out. Here for a while. Pay for to get out after this.
I just well, one we have
So
When did you graduate? 3 years ago.
in what? MMI?
Yeah. I was in direct. Yep. Jerry's awesome.
So we were actually in Boston first because she moved up there when I was still there.
I see.
We swapped places basically. I became a Harvard postdoc and she became a college. She's just always more.
Well, you know what your next job is then.
Apparently. That means she needs to apparently no city can handle the 2 of us for very long.
Yes, we have a game tomorrow.
I'll do it.
And of course, I already told Lindsay, when your defense is, she could like not let me know. Like, God damn it, what is your defense?
Secretive, aren't you?
Yeah, and I'm like,
You were in Australia being busy.
yeah, that's true. And then I kept forgetting, and then I was like, oh, sweet. But then my flight was like a different flight was coming tonight. I was like, oh, so check that thing.
So that's my.
Oh, this was the Australia thing that you did last year.
Or yeah,
2 years ago. Yeah.
yeah. So
How did that go?
it was class.
Yeah, awesome.
No injuries, not for the 2 of us. For other people, yes. Do we know how Greta's doing surgery?
I don't think so. She's back in the US.
Okay. Just why they have to do surgery because they don't actually grow and progress anything. And I was like, how can she fly home?
Oh, yeah.
Oh, well. Yeah.
Yeah, that's such a good start.
It was real nasty. I was like, yeah, because they don't have to have ambulances at like fields or anything. And I was actually talking to a guy about that. And he's like, yeah, actually there's a shortage in paramedics and whatnot that goes over there. So
I saw a video. Yeah, you know, she was sedated, so she doesn't remember it, but they were trying to get it.
So tired working.
Yeah, it's really cool.
yeah, apparently they what, like put her under like
So
slightly.
that time they failed.
Yeah, and they moved it, and then she like woke up and like screamed, and then like went back to sleep.
You don't want videos of this. I think you just don't want to have videos, right? It's like,
I think Lindsay was the video, right? I was kind of abusing.
People post it.
Yeah,
I think Ali asked for the videos, but
maybe.
That's okay, I'm on an island.
Yeah.
He did just join like.
Yeah. You're good.
so you're gonna be filming it there?
Then don't be silly like Michael's and stand right in front of the camera.
Yeah, there's a video of his because I did the
And
same
you see
thing.
what you see, like.
Well, yeah, so like.
I just saw his crotch for like a very long
That
period
was
of
in the,
time.
that was in the Q&A. So.
Yeah, but still, like I didn't want to be
Because they
staring
do
at his
it,
crotch.
they have to do it in the summer hall or something. So you're on a stage.
Right.
So the computer's down there and you're up here. So when you walk in front of it.
Yeah, I didn't think he decided that was the spot.
No, it looks like if I walk in front of
And
it,
I
I'll
was
just
like
be.
interested in the Q&A, so yeah, that was fun.
Probably just cover behind me.
That's that, yeah.
Oh, you'll hear you.
So Brendan, who else?
Yeah.
Andrew.
I just bumped them over
That's
my.
rolling.
Well, I was in the car. Brendan drove me in today, so.
Yeah, he's the one I think is most likely to.
Good yesterday.
There are a lot of smiley faces in your car.
And where's the Q&A? It's gonna be good.
Yeah.
32 days.
I warned Alex yesterday. I said, you can tell your girlfriend I'm not usually drunk, but I'm really.
Oh, it's gonna be a polygraph, and I'm really excited.
Okay, Brendan made it.
Yep.
Oh, I just haven't seen it. But I
You
can
guys
try
can
it.
stay around a little bit and party out.
Yeah. You guys are doing like a questionnaire first, right? Don't you guys like take it away?
No, not after this. It's a presentation and then public questions and
Oh,
it's not a
sweet.
private anything.
Sweet. I don't know, like, I don't know, you have to like, take it like an hour.
Yeah, somebody
So, like, really
This is basically.
It seems like I talked to like.
awesome.
Sorry.
They don't have to grill you, I didn't know that. Awesome.
You just have to like,
Well, that is so much better than that. Yeah.
you know,
Partially because I did this program, so clearly they just all the different. You really big? That's nice.
I'm pretty glad you were there because you totally would walk my letters.
Oh, shoot.
Yeah, I gave it.
That would be funny. That would be really amusing. We should explain that those are not birds, those are actually chromosomes. I don't know. I was like,
If I say that,
like it's either or.
Yeah, just some sort of weird abstract.
it's like that psychologist test.
Mhm.
What does this look like to you? That's what that chromosome typing stuff is. It's just
Well, welcome everybody. I want to welcome you to Alex's thesis defense. It's really with great pleasure that I have a chance to introduce Alex. Sorry, gonna start again. These are really important occasions to mark. Anytime there's a really significant event, it's great to come together and really have a chance to celebrate. And for me, I think this is one of the most rewarding things that I really do by far in my job. The connection with young scientists, which really is a two-way learning event. And it's the best thing about teaching because it goes both ways. While Alex may have learned some things from me about polymerase processivity or the fine art of filleting a paper in Journal Club, I've also learned a lot from her. Alex taught me about the power of individual action. As many of you know, Alex is committed to the environment. I've been green at heart for a long time, but I wasn't always so successful at executing that. By example, at lunch, she always used a real plate and real knives and forks. No paper, no plastic. Green for the environment. And she quietly set the example for all of us. It took me some time to catch on, but then I got it. And now for parties at my home and for lab lunch, we have real plates and real paper. Real plates and real silver. No paper or plastic. All right, it's the drugs, guys. I'll explain. Well, Alex changed our behavior to green in a very quiet and gentle way. That does not mean to say that she is always quiet and gentle. One of the traits that Alex brought with her to the lab, and that we actually share, is her attitude of work hard, play hard. So it's not surprising that when Alex fractured her collarbone playing footy hard last year, we both took it in stride and found workarounds for moving her thesis forward. And then I followed in her footsteps and fractured my finger hard playing boogie boarding. And now again, we're taking it in stride, except for the drugs that make me talk funny. It seems that whatever synchronicity that we are in, Alex is leading. So I wish her an easy and healthy journey after her PhD and whatever happens in her life. Because apparently what happens to Alex with some delay will happen to me. Alex might have learned once, twice, or several times during the time in the lab to believe in herself. This is an important lesson in science. Experiments have a way of always throwing you a curveball, but never the one that you expected. I always believed in Alex, and Alex came to believe in herself too. When data are confounding, and apparently not the same as the day before, we would go together and look for the reason why. So today, you will hear the sum of the points that we arrived at. These will be the conversations in her talk today. And the work that Alex will present is very impressive. But trust me, the behind the scenes work that led to these conclusions was much more than you would imagine. It's been a pleasure to walk this rocky, curvy road with you, Alex. And I look forward to more playing hard together.
Thanks, Carol. That was quite the introduction. I feel honored to be up here and having taught Carol Wright or something in my time here. It's a good thing. So thank you all for being here. I'm excited to present to you the work that I've been doing over the past few years. And I just wanted to start out with this slide that is covered with what most of you know are metaphase chromosomes from human cells. All of our genetic information is packaged into these little chromosomes, and the cell does a lot of work to keep them safe. There are a lot of different mechanisms in order to keep that going and protect your genetic information, but the part that we're most interested in our lab is the telomere. And of course, that comes from the Greek, telos meaning end, and meros meaning part. So telomeres are the end parts of the chromosome that help protect it from being recognized as damaged DNA, and also protect it from what's known as the end replication problem, where the telomeres lose a little bit of sequence every time a cell divides. And that's because of something inherent with the mechanism of DNA replication. So here's just another cartoon of a chromosome. The telomeres are here in blue, and I have to note this is not to scale. I've enlarged the telomeres for your viewing pleasure. So the telomeres in humans are a repeated sequence of TTA-GGG, and that covers about 8 kilobases. And most of that sequence is double-stranded DNA, and ends in about 100 nucleotides of single-stranded DNA at the end. So the telomere is also coated with a protective structure, or with a complex of proteins called shelterin, and the shelterin complex helps protect the telomere and regulate telomere length. So shelterin consists of 2 sets of double-stranded DNA binding proteins, TRF1 and TRF2, which bind the double-stranded TTA-GGG sequence, POT1, which binds single-stranded telomere sequences, and then these accessory proteins TIN2, TPP1, and RAP1, which bind to telomeres specifically through the DNA interacting proteins. And shelterin has several major roles, including preventing the ends of the chromosomes from being recognized as DNA damage, and also positively and negatively regulating the enzyme telomerase and its activity at the telomere. So I mentioned to you that a little bit of sequence is lost from the ends of chromosomes every time a cell divides. And that's all right because it's repetitive sequence, it's not coding for anything important, but after many rounds of replication, telomeres get critically short and lose their protective ability, and then become recognized as damaged DNA, which leads to senescence or cell death. But the enzyme telomerase is there to help recover these telomeres back to an equilibrium length by adding de novo telomere repeats from its RNA template, from the telomerase RNA. So a telomere isn't a set length, but it's actually maintained, the telomere length is maintained as an equilibrium distribution. So there's a lot of different lengths centered around mean telomere length. And as the cells divide, you lose a little bit of sequence from all the chromosomes. And if that's not corrected by telomerase, you end up shifting the equilibrium to shorter telomeres, and then that leads cells to die and undergo senescence, so that's bad. And telomerase exists to elongate, and preferentially elongates the short telomeres to restore the telomere length distribution. So this is important actually for cells that need to divide many times. So in normal tissue renewal in your body, you have a lot of stem cells that need to continuously divide throughout your lifetime. So normal tissue renewal depends on telomerase to maintain the telomeres of those cells. And without telomerase, you can lose tissue renewal, leading to age-related degenerative diseases. And the converse of this is in cancer, that's a cell that's dividing more than it's supposed to. So telomerase is activated in 90% of human cancers, and that's bad. So you need to telomerase is very highly regulated to achieve a balance between dividing enough times to fulfill your functions and cancer. So in addition to the heterogeneity you see in chromosomes or telomere links in an individual cell, you also see a wide range of telomere links in a population. And what's shown here is just a graph of a lot of individuals and showing their telomere length over their age. And I want to point out two things about this graph. And first is that telomere length decreases with age. And second, that telomere length is very heterogeneous. So that means you can see a 25-year-old and a 65-year-old might have the same telomere length, and they're all perfectly healthy. So what people have done is taken a lot of measurements of telomere lengths in individuals and put an upper and lower limit of the normal telomere lengths so you can identify clinically relevant telomere length patients. So what that looks like is the same plot here. These are the percentiles. So the top in red is the 99th percentile and the bottom is the first percentile. Most people fall somewhere in the middle and they're perfectly healthy. But once you get around or below the first percentile, that's when you start having short telomere syndromes, as we call them. And that's a heterogeneous number of diseases who share the etiology of short telomeres. And so some of these telomere syndromes include rare diseases like dyskeratosis congenita and some bone marrow failure syndromes, as well as a more common disease known as pulmonary fibrosis. And studying patients with these short telomere diseases helped us learn a lot more about how telomerase is regulated in human cells. Many short telomere patients, their short telomeres can be traced back to mutations in a number of different genes related to telomerase or things that regulate telomerase. And a lot of these are in the telomerase holoenzyme itself or factors involved in biogenesis and stability of telomerase. And we know that telomerase is haploinsufficient, meaning that having half the normal level of telomerase is not enough to maintain telomere length in a person. So when I started this project, we got really interested in mutations in shell-turn proteins that showed up in some patients with telomere disease, and it wasn't really clear how these mutations were causing extreme telomere shortening that we saw in patients. And when I started, TIN2 was the first shell-turn protein observed mutated in short telomere patients, and later TPP1 showed up also mutated. Another interesting thing about these patients is that a lot of telomerase loss of function mutations show up in families with genetic anticipation. So the telomeres gradually shorten over multiple generations. But many of the TIN2 mutations actually cause rapid telomere shortening. So here's a similar telomere length diagram. And these red dots are unrelated patients with TIN2 mutations. And in blue are their siblings and here are their parents. And so they clearly went from normal telomeres in one generation to extremely short telomeres and very severe disease. So we were also intrigued how that could be happening. So a little more about the TIN2 patient mutations is the mutations are often de novo, meaning they're not inherited from either parent. And the patients are heterozygous for the mutations that are either missense or nonsense, so a single change in an amino acid or a truncation. And it appears that all the mutant proteins are stable. So this indicates that it's some sort of dominant mechanism that these TIN2 proteins have to cause disease. So what we know about TIN2 function is that TIN2 interacts with TRF1, TRF2, and TPP1, and it's important for their stability at the telomere. If you get rid of TIN2 by shRNA knockdown or knockout, none of the other shell-turn proteins can localize to the telomere, and you end up getting a DNA damage response and cell death. And you can also see a slight telomere elongation. So TIN2 is thought to be a negative regulator of telomere length. But the patient mutations they maintain their binding interactions with TRF1, TRF2, and TPP1, and have normal telomeric localization. So it's not acting through any of the known functions of TIN2. Other groups have been studying these TIN2 mutants as well, and some have pointed to, or some have observed that there's a reduced association of telomerase with telomeres in cells, but other people have reported telomerase-independent telomere shortening. So it's still not clear what the mechanism of telomere shortening is in patients carrying these TIN2 mutations. So this is a schematic of the TIN2 protein. There's not a lot of information we know about it. So TIN2 is only found in vertebrates, and it has no known domains or homology to other known proteins. And other people have mapped out its interaction domains with TRF2, TPP1, and TRF1. And this red mark right here is where all the patient mutations cluster. From multiple sequence alignment, we've seen that it's pretty conserved across the protein, especially in the N-terminus where it interacts with TRF2 and TPP1. And the TRF1 binding motif is pretty well conserved, as is the patient mutation cluster, indicating that this is an important functional part of the protein. We just don't know what it is. So from amino acids 269 to 290 is where almost all of the TIN2 patient mutations are clustered. And the disease severity depends on which amino acids are mutated. I have marked K280 and R282, and those are the most severe mutations. But you get disease when basically anything in this cluster is mutated. I'd also like to point out that TIN2 has two expressed isoforms, TIN2 short and long. And that's in contrast to the mouse that only has one isoform that's more similar to TIN2 long. And we also notice there's a patch that's also highly conserved in the C-terminus. That's suggesting that maybe there's something relevant about TIN2 long, but all the studies trying to understand what TIN2 is doing to cause telomere shortening have been done in either TIN2 short or in the mouse TIN2. So we, throughout this project, we're going to look at the different isoforms of TIN2. So the two isoforms result from alternative splicing of the TIN2 gene, which contains 9 exons that are either all spliced together to form TIN2 long, or they retain the last 3 introns, leading to a stop codon right after exon 6, and that's TIN2 short. All the patient mutations are clustered in exon 6, so both proteins will have the patient mutations. So we were wondering which isoforms are expressed in human cells and whether or not they were in equal levels, and do they do anything different? So not much has been described about the differences between the two isoforms. And answering some of these questions has been difficult because there's not we couldn't find a very reliable commercially available TIN2 antibody. And they're hard to tell the difference between by qPCR assays because these retained introns are pretty small, so it's hard to tell the difference between the transcripts. So what we decided to do was do a CRISPR knock-in of an epitope tag at the N-terminus of the TIN2 gene, and that way we could follow the proteins by Western blot. Guide RNA targeting right by the start codon of the TIN2 gene, and inserted a MIC tag at the endogenous locus. So when we did a Western blot on some of these CRISPR clones, we saw that there were 3 MIC bands instead of the 2 that we expected. And we used cDNA of TIN2 short and TIN2 long and realized that the unidentified band is somewhere between the 2. Because of the size difference, we thought that it might be a new isoform of TIN2. So we wanted to look at the RNA and see if there's another isoform. So we did a modified 3' race by taking the RNA from human cells and reverse transcribing it with the oligo-DT primer with an adapter, and then PCR amplifying the cDNA with a primer in the exon 1 and a primer to the adapter sequence. And then using PacBio sequencing, we're able to get long read sequences that go all the way across the transcripts. So then when we got that data back from the HIT core, they took the CCS reads and filtered for full-length reads, and we used HiSat2 to align them to the genome and use string tag to model gene populations to see what the TIN2 expressed isoforms are. And there's several observations that we saw. So in the human cell lines, we saw that there are many different splice variants and lots of examples of intron retention and exon skipping. Some of those were common to all the cell lines we looked at, and some were unique. And there were several incidences of alternative polyadenylation. But we observed 3 major isoforms, including TIN2 short, TIN2 long, which we expected, and then this third one, TIN2 medium, which would be translated into something similar to the size that we saw in our Westerns. So TIN2 medium basically just retains the last intron between exons 8 and 9. And it has most of the same sequence as TIN2 short and long, but codes a little bit of unique sequence from the retained intron. We saw TIN2 medium in 5 different human cell lines and not in either of the mouse cell lines we looked at. which is consistent with what's in the literature about mouse only have one expressed isoform. Mouse also had fewer examples of these abnormal splicing events. I've also looked in a publicly available isoseq data set, so that's the same PacBio sequencing, but genome-wide. So this is PCR amplified, so we can't really say anything about the ratios, but this publicly available data set also had TIN2 medium. And we also looked at a ribosome profiling database, and it supports that TIN2 medium might be translated. So we solved a lot of questions about the differences between the isoforms, and now we have a new isoform to ask those questions about. And we've done a little bit of work on this. And first, we checked to see if the TIN2 is still localizing to telomeres. So we just overexpress the different TIN2 isoforms in HeLa cells. And when you stain for TRF2, that stains the telomeres, which you can see as little dots. And MYC is marking the TIN2, and those dots co-localize nicely with TRF2, which is to be expected because all three isoforms have the binding domains for the other telomere proteins. But they're all localized to the telomere, and expressing one of the patient mutations, K2AE, in all three isoforms still localized to the telomere, which agrees with data in the literature that the patient mutations don't affect localization. So we're also looking into whether these affect telomere length differently and how they behave in telomerase assays, which I'll get into later. So to summarize this first section, TIN2 is expressed in three predominant isoforms, TIN2 short, medium, and long. And the mouse only has one isoform that more closely resembles TIN2 long. All three isoforms localized to the telomere contain the mutation cluster. And most of the previous work, as I mentioned, has been done in either TIN2 short or in mouse TIN2. So we saw the questions of how are the isoforms behaving differently, and that's to be determined. We also have the questions, how does TIN2 regulate telomere length? And do the different isoforms have different effects? So to address this, we turn to TIN2's interaction with TPP1. So TPP1 is part of the TPP1-POT1 processivity complex for telomerase. So TPP1 interacts with telomerase through its TEL patch, which touches telomerase and brings it to the telomere and stimulates its processivity. TPP1 also interacts with POT1, and they function as a heterodimer in cells. And POT1 binds the single-stranded telomere. So we wondered if TIN2 could be affecting telomerase through its interaction with TPP1. So before I go on with that, I'd like to explain a little more about how telomerase adds telomere repeats. So here's a telomere seed sequence, and this is the telomerase enzyme. And it uses its RNA template to align to the end of the chromosome. And then it fills in from its template a telomere sequence. And then once it's finished filling in a telomere sequence, it can either translocate or fall off. And so it'll translocate and add another sequence. So the number of times that telomerase translocates and adds more sequence determines the amount of telomeres added. And so we're defining processivity as the average number of repeats added in a single binding event. And changes in processivity are sufficient to cause disease. So there have been mutations in TERT, which is the telomerase reverse transcriptase component, that specifically affect processivity of the enzyme and not its ability to add nucleotides. And those are found in patients with short telomeres. There have also been mutations in the TPP1 TEL patch that cause reduction in processivity telomerase and cause disease. So to measure telomerase processivity, we start with a telomere primer and then provide telomerase either recombinantly expressed or from cell lysates. So I do it in cell lysates, and you give it the nucleotides it needs and also radioactive GTP so you can follow the products that incorporate it. And you let telomerase add to the primer, stop the reaction at different times, and then separate the products on a sequencing gel. And so what that might look like here in this cartoon, the first lane is a processive telomerase, and the second lane is a less processive telomerase. And the more processive telomerase is going to have a higher number of high molecular weight products versus the less processive that are going to be shorter. And you can quantify this by measuring the intensity of the bands at the top of the gel and comparing it to the bands at the bottom of the gel. So this isn't a direct measure of telomerase processivity, but others have shown that it corresponds to the processivity of the enzyme. And we know that changes in processivity can cause disease. So in this 2012 paper by J.K. Nandakumar from the Czech lab showed this is what they used to define the TPP1 TEL patch. And they used cell lysates to test telomerase activity. So you can see this is what the data looks like, not my cartoon, but in the first lane, they just overexpressed telomerase by itself. You can see there's not much in the high molecular weight products, but most of the products are lower molecular weight. When you add TP1 POT1, you see a lot of bands that are high molecular weight compared to the bottom. And so when you quantify it, it looks like this. And you can see that TPP1 has about twice as processive when TP1 POT1 are there. So then they use mutations in the TPP1 TEL patch, or they use mutations to define the TPP1 TEL patch. And so that looked like this. So some mutations didn't affect processivity and some reduced processivity, and you can see that by looking at the ratio of the high molecular weight bands to lower. So we thought this system would be a nice way to test whether TIN2 affects telomerase processivity by just overexpressing these components in cells and adding TIN2. So we can ask, do the TIN2 mutants affect telomerase processivity? And asking the question, does TIN2 act through TPP1 POT1 by mutating the TEL patch? So to test this, we created some telomerase assay cell lines where we could hold constant TPP1 POT1 and telomerase and add different TIN2s and test whether or not they affected telomerase. So to overexpress TPP1 POT1 and telomerase at similar levels, I stitched together with 2A peptides, TP1 POT1 and TERT to create a single polycistronic expression cassette to try to get the proteins overexpressed at similar levels. I also made a cell line with the TEL patch mutation or with TERT by itself with no TP1 or POT1. And all of three of those proteins are flag tagged, and you can see them on a Western blot. So there's TPP1, POT1, and TERT. And the expression cassette expresses all three of them, and a couple extra bands that we think might be telomerase degradation products. So generating these cell lines, we started with 293 T-Rex flippin cells and transduced them with a telomerase RNA-expressing lentivirus and selected clones that expressed high levels of telomerase RNA, and then used the flippin system to flip in these different constructs to make 3 different cell lines. So that's what these cell lines look like on a Western blot. So the TERT-only cell line is expressing TERT, and the others are expressing all 3 proteins. And we have noticed that there's less telomerase overexpression in the TERT-only cell line. We haven't been able to figure out if that's an artifact of this expression cassette or if overexpressing TP1 POT1 is stabilizing TERT in some way. So when you do a telomerase assay on these cell lines, this is an example of an assay stopped at 10, 20, or 40 minutes in the TERT-only cell line. You can see the increase in high molecular weight bands with time. And then when you use the TP1 POT1 cell line, you see these higher molecular weight bands aren't present with TERT by itself. And then the TEL patch mutation cell line, some of these higher bands go away, and they're actually in the bottom of the gel, so they're shorter, making shorter products. And we've used several different ways to introduce TIN2 to these cell lines. So first is transfecting the TIN2 full-length gene. Conveniently, it's about 3 kb, so you can transfect it pretty easily. Or transfecting cDNA constructs of the 3 different isoforms, with or without patient mutations. And also with our collaborators, we've been adding recombinant TIN2 long to the telomerase assay lysates. So I'm going to start with no, I'm not there yet. So to make sure that TIN2 is forming a complex with telomerase, we did some co-IPs. So TIN2 is MIC tagged and all the other components are flag tagged. So just when we did a MIC IP, we saw co-IP of telomerase, POT1, and TPP1. And then we did the reciprocal IP with flag and saw that we were able to pull down all isoforms of TIN2. So the first thing I'm going to show you is a transfection of the TIN2 full-length gene. And we transfected it into the TP1 POT1 cell line and transfected either wild type or patient mutation, as well as GFP as a negative control. And we saw that both the wild type and the mutant expressed all 3 TIN2 isoforms. And when we did these telomerase assays and stopped them at 5 or 10 minutes, we were actually really surprised to see that compared to the GFP cell line, the TIN2 full-length wild-type cell line stimulated telomerase persistivity. You can see these bands that are not present in the GFP lysate. And then in the lysate with the patient mutation, those bands disappear. And they're actually slightly worse than not having TIN2 overexpressed at all. So this was exciting. We thought that maybe the patient mutations would be reducing persistivity, but we didn't expect that adding TIN2 itself would stimulate persistivity over the TPP1-POT1 stimulation of telomerase. And this is another piece of sort of preliminary data from collaboration with the Wookiee lab at Boulder. So they sent us some recombinant TIN2 long purified from E. coli, and we added that to our telomerase assay cell lines. And first, we wanted to make sure that the recombinant TIN2 long could, in fact, interact with telomerase. So we did another IP, and TIN2 long did come down with the telomerase complex. And when we added increasing amounts of this recombinant TIN2 to the TPP1-POT1 lysates, we saw that, again, it stimulated persistivity. So these two pieces of preliminary data suggested to us that TIN2 stimulates telomerase. We saw that in the TIN2 full length, and also with recombinant TIN2 long. So we wanted to further characterize this, and we asked, does TIN2 actually stimulate telomerase? And is this dependent on TPP1-POT1? And do all the isoforms behave the same way? So to address this, I transfected all 3 TIN2 isoform cDNAs into either the TP1-POT1 cell line or the TP1 TEL patch cell line. We did this in triplicate to do telomerase assays and see if these isoforms can stimulate. And so what that gel looks like is here. And this is an assay that's gone for 40 minutes. And I have to apologize, in this lane, I lost the sample, but I have another gel with that there. And so what you might be able to tell is that some of these are darker than in the GFP cell line, where we have TIN2 short, medium, or long. It's really hard to tell by eye, so we quantify this and see about a 10 to 20% stimulation of telomerase in transfections of TIN2 short, medium, or long. So then the question is this TIN2 stimulation through TPP1 and POT1? So I did the assays on the TEL patch cell line. And you can first of all see the TEL patch cell line is much less persistive. You don't have these high molecular weight bands. But we didn't see any change in persistivity in those cell lines. I've also done this in the TERT-only cell line, which has no overexpressed TPP1-POT1, and we didn't see much of a difference there either. So we conclude that this TIN2 stimulation of telomerase requires TPP1 and POT1. So to summarize this, we see that TIN2 is cooperating with TPP1 and POT1 to stimulate telomerase persistivity. And I know that the stimulation of telomerase seems subtle, because you can see about a 50% increase in telomerase activity with TPP1 POT1 and about 15% with TIN2. So what we're thinking is, although there are a lot of different ways you can change the activity or persistivity of this enzyme, we think that TIN2 is completing the TPP1 POT1 persistivity complex and either creating more fully assembled processive telomerase complexes or favoring the TPP1 telomerase interaction or the POT1 single-stranded DNA interaction, or both, to create more processive telomerase activity. And so we've already known that TIN2 is required for TPP1 POT1 function in vivo. So in cells without TIN2, TPP1 POT1 don't go to the telomere, and TPP1 doesn't recruit telomerase to the telomere. So we already know that it's important for this complex. And we've seen TIN2 stimulate telomerase persistivity in several settings. And we're starting to think of this TIN2 TPP1 POT1 complex as a heterotrimeric single-stranded binding complex, which I'm going to talk a little bit more about what the implications of that are. So when you think of single-stranded binding complexes, most people think of RPA, replication protein A, which is the global single-stranded binding complex in a cell. And RPA coordinates DNA replication, DNA damage, and repair. It's a heterotrimer that consists of RPA 70, which is its large subunit and has its main contribution to DNA binding through its OB folds A and B. And it can also engage these other two OB folds, B and C, to have a larger footprint on the DNA. And RPA, through its interaction with single-stranded DNA, recruits and coordinates the activity of other enzymes during these processes of DNA replication and damage and repair. Telomeres have some specific needs with their single-stranded DNA, particularly telomere replication is kind of challenging because it's a very repetitive sequence and it's very G-rich. So it needs a little bit of help getting the replication forks through the telomere. Telomeres also have this in protection necessity to mark the natural end of the chromosome and protect it from being bound by RPA, which will trigger it as a damaged DNA, and also regulate telomerase activity at the end of the telomere. So there's a couple of telomere-specific single-stranded binding complexes that I'm going to talk to you about. And the first is CST complexes, which are also known as tRPA. So it's a telomere-specific RPA. And they're conserved across organisms and bind either telomere DNA specifically or just G-rich DNA. And they share the common function of stimulating DNA polymerase alpha, which is lagging strand polymerase that primes and replicates the lagging strand. CST was first identified as a tRPA in yeast, and it looks very similar if you look at the schematic to RPA, but it has some interesting differences. In that CDC13 is the large subunit, and it only uses one of its OB folds to bind DNA. And STEN1, which is the middle subunit, has two winged helix domains instead of one. And yeast CST has some additional functions on top of stimulating Pol alpha. It both serves as an in-protection complex in yeast and also recruits telomerase. The large subunit of CSTs is more divergent. So in tetrahymena, it's much different. But tetrahymena CST is also telomere specific. And then in humans, the large subunit is not telomere specific, but is actually specific to G-rich DNA. So that it goes to telomeres, but also to other hard to replicate sites in the genome. There's also a class of telomerase processivity complexes, including this P50 TEB complex in tetrahymena, which consists of P50 that interacts directly with telomerase, kind of the way TPP1 does, and then TEB1, which binds telomere DNA, and TEB2 and TEB3, which resemble RPA proteins. And I've told you quite a bit about POT1, TPP1, and TIN2, but this complex, POT1 binds the single-stranded DNA, and TPP1 interacts with telomerase. So what's interesting is that both the telomerase processivity complex and the CST complex in tetrahymena are part of the telomerase holoenzyme that a group isolated and solved the cryo-EM structure for. We're wondering if coupling these two single-stranded binding complexes is conserved in humans as well. So this is a cartoon of the cryo-EM structure of tetrahymena telomerase. And this section in the middle is the telomerase holoenzyme core. And you can see P50 right here in red. And this is the TEB complex, and here's CST. And so it was very surprising to find both of these single-stranded binding complexes as part of the tetrahymena telomerase holoenzyme. It was also interesting that TEB1 was the only part of the TEB complex that was known before the structure was solved. So these smaller components, TEB2 and TEB3, were not identified until this group solved the cryo-EM structure. And TEB1 was known to stimulate processivity of tetrahymena telomerase, which you can see here in a telomerase assay without TEB1. And then with TEB1, you get a lot of processive telomerase activity. But then if you have the full TEB complex that includes TEB2 and TEB3, you get an even greater stimulation of telomerase than you get with just adding TEB1 alone. And this reminded us of what we saw with TIN2 stimulating telomerase processivity above what we knew TPP1 and POT1 were able to do. But in addition to the processivity aspects, this structure gave us some new insights about how telomere elongation is coupled with telomere replication. So the group that did this also mapped the path of the single-stranded DNA across TEB1 and into the telomerase template. So the end of the DNA is going to go in through there, but also as telomerase adds repeats, it's going to be extruded back through TEB1. And so you can imagine drawing the rest of the single-stranded DNA that it might loop back around and go through CST. which is going to stimulate polyalpha primase to complete lagging strand replication of the telomere. So this coupling of the G strand synthesis by telomerase, adding TTA, GGG, and the C strand synthesis of the lagging strand, has long been it's been known. So it's been observed in yeast that if you don't have lagging strand polymerases, you don't get telomerase elongation. But it's never been understood how that might work. So this provides insight to that coupling. And so we're thinking of single-stranded binding complexes that playing two roles at telomeres. First is stimulating telomerase processivity, which I've mentioned to you many times, by TIN2, TP1, or PA1, and also facilitating replication fork progression, not only by CST stimulation of DNA polymerase alpha, but also by the TIN2, TP1, PA1 complex. And this, there's some evidence indicating that it might be the case. So PA1 mutants lead to replication stress, which is indicated by excessive telomeric single-stranded DNA and replication fork stalling at the telomere when you have PA1 mutants. So we're thinking that while a lot of a lot of single-stranded binding proteins of the telomere are often drawn at the end, they also have an important role at the replication fork. In addition to that, we think that replication fork progression through the telomere and replication fork collapse are important telomere length regulating processes. So this is an illustration of the replication fork, and as it gets towards the end of the chromosome, have the telomeres here in blue, you switch from having RPA bound to having telomere specific single-stranded binding complexes. And that's going to help when inevitably the telomere or the replication fork collapses, or the replication fork stalls and then collapses. You're going to have two strands of DNA that aren't done being replicated. So the leading telomere is going to have a leading strand polymerase on it, and it might just be able to be completely replicated when that happens. But the lagging strand telomere is going to need a little bit of help, or you're going to lose all of this sequence. So that's where these telomere single-stranded binding proteins come into play. So they can stimulate polyalpha primase to finish Okazaki fragments in the telomere and also stimulate telomerase, leading to complete telomere replication with the 3' overhang. But if you have defective telomeric single-stranded binding proteins, this lagging telomere is not going to get finished, basically. So you're going to get it coated with RPA, which can signal a DNA damage response and inappropriate DNA repair. But and what you'll see also is increased telomeric single-stranded DNA, and you'll have a net sequence loss because this complementary strand was never finished being made, which will show up in the next round of DNA replication. This becomes the new telomere and it's truncated. And you might also see defective telomerase addition, but this telomere truncation is going to overshadow that. So this is what we think might be happening in some of these TIN2 patients that have extremely short telomeres. So I'm just bringing back up a slide that I showed you at the beginning, where many telomerase loss of function mutations cause gradual progressive telomere shortening over multiple generations. But in some of the telomere patients, you see rapid telomere shortening in one generation. What this would look like as a cartoon is many rounds of replication without telomerase activity will gradually give you short telomeres, and that's a result of loss of telomerase activity or processivity. But if you have telomeric fork collapse that's not repaired, you'll lose large pieces of telomeres and get short dysfunctional telomeres much quicker. So of course, all of this needs to be tested. So that's the future direction, is testing whether the TIN2 mutations have a role in replication fork stabilization and replication fork collapse. To summarize, we found that TIN2 has multiple expressed isoforms, 2 that were already described and a new one, TIN2 medium. And we're working on understanding the effects of the different isoforms because that's not yet known. We've seen that TIN2 cooperates with TPP1 and POT1 to stimulate telomerase processivity. And we're starting to think of TIN2, TPP1, and POT1 as a heterotrimeric telomeric single-stranded binding complex. And so the patient mutations have the patient mutations have unexplained telomere shortening because it's greater than loss of telomerase itself. And so we're thinking that Telomere SSBs may also have a fork stabilization role, and disrupting them can uncouple telomerase from telomere replication. There's a lot of future directions. To finish examining the roles of different TIN2 isoforms in human cells, and further define the effects of the patient mutations on telomerase activity and persistivity, but also to test TIN2's role in telomeric replication forks to see if what we're thinking is true. With that, I'd like to thank a lot of the people who've helped out with this project. Carol, of course, Margaret did the IF that I showed you. Carla helped me learn telomerase assays. John Paul helped with some of the PacBio sequencing in his rotation. The Armanius labs and Holland labs for their support. John Alder helped with the CRISPR experiments. Our collaborators, Debbie Wicke and Leslie from Boulder, sent us recombinant TIN2. Of course, the sequencing facility, Sarah Whelan helped with the data analysis, my thesis committee, CMM, and chief. With that, I'll take any questions. To increase? I guess we'd have to further define how TIN2 is interacting with the processivity complex to understand, because I guess TIN2 overexpression could simply, do that, but we don't understand the mechanism of how TIN2 works. But yeah, it could do that.
Longer telomeres, maybe.
Yeah, well, yeah, it could lead to telomere elongation. The isoforms might also have different effects.
So do you see, clinically, do you see this rapid collapse in POT1 and TPP1 mutation, or is it only in the TIN2?
So I think it's only in the TIN2. The TPP1 patients have short telomeres, but I don't know what their family members look like. But definitely TIN2 was the first one where you saw that fast telomere truncation. And it could be that, so POT1's confusing too, because if you get rid of it, telomeres get longer. But that could be an artifact of measurement, because it could be that you're reading longer single-stranded DNA. But there's also mutations in CST components in humans that cause Coats plus disease, which is either a replication disease or a telomere disease. Some patients have short telomeres and some don't. But yeah, I'm sure.
So what I'm wondering is, if TIN2's function, I mean, can you? I really like the replication fork collapse model, but I'm wondering if you could suppress that. Like, could you suppress rapid collapse by knocking out additional telomerase components, like a TPP knockout in the background of a TIN2? Would that actually be?
Sounds like you're a eugenicist. Yeah, that would be interesting, right? So we could easily just throw in an SHRNA or something to TPP1 and see if that rescues it.
I mean, do you think you can model this in just in cell culture with a knockdown or turn off of TIN2?
So it's confusing because I think a lot of the sheltering proteins have been studied by knockdown and overexpression, but their endogenous levels are very low. So I think the stoichiometry is really important. So you get confusing things with knockout. So if you knock down TIN2, the cells die because the telomeres just start signaling DNA damage. So they don't grow for very long. And if you can get them to grow for a little bit, telomeres get longer. Which is part of why the telomere shortening in patients was intriguing, because it seemed like they would get longer if you got rid of TIN2. So it seems like the TIN2 mutations are not inactivating mutations, it's that they're affecting a functional domain.
But maybe you could knock down TIN2 in that exact model in a TPP1 knockdown in the background.
Yeah.
Without the cell dying. Maybe it's dying for exactly the reason here.
Sure.
Anyhow.
Yeah. Well, it could be worth looking at. Yeah. Anyone else? That's it.
I was just going to ask, maybe this is along the same line. So if you theoretically uncoupled the replication role and then the role of stimulation across acidity, you would see, if you only had the stimulation, you would see gradual shortening with immune potentially.
Right.
You wouldn't see this drastic shortening.
Yeah. Well, so that could be the case with the TPP1 mutants, right? Is that the TPP1 patient mutations are specifically in the tau patch. So the rest of the complex is still there, but it's only really affecting the TPP1 telomerase interaction. But maybe this TIN2 domain is, yeah, I don't know. I don't know if there's a domain of TIN2 that you can get the separation of function, but it's possible. Yeah. If there's no more questions, I'm gonna do some more acknowledging of people who've been involved in this project. Yeah. Yeah, so I did thank some of the people that were involved in specific parts of these experiments, but there's a lot more than that. So I really need to acknowledge the Grider Lab experience in making all of this work possible and keeping my sanity over the past 6 years. So it's been a really fun lab to be a part of. You know, we like long walks on the beach, long bike rides on the beach, advocating for important things like science in general. It's been a really fun and supportive environment. Place that really allows you or compels you to work hard in the lab, but also lets you balance that with work hard, play hard, like Carol brought up at the beginning. And some of the specific people that have made this experience really great. Carol, couldn't have asked for a better advisor. I'm happy to hear that she's learned a few things from me along the way, but I've learned a lot from her. And I don't even know how to express how grateful I am for all of the data meetings that we've had, interesting discussions and encouragements when things don't work as you expect they might. There's a lot of ups and downs in grad school. In addition to the learning techniques that you're working with, you get surprising results, which end up being interesting, but it's quite a roller coaster. And what really makes that worth it is when you send your boss some data and she accidentally saves it as the background of her phone. So you feel like you've accomplished something. So yeah, Carol also gives good advice to help you get through things, and I've heard her quote Chumbawamba, get knocked down and get back up again, and that's something that you do in the lab and also in life. Carol's fun to dance with. But also I have to say that we've all gotten down and gotten back up again. So these injuries that Carol mentioned are not the only ones that have happened. You're in the lab. But you keep going, you keep dancing, you go boat, you bike around the lab and, you know, carry your beers around in your bike basket. I don't know, it's been great, and I can't thank Carol enough for everything. But this is really the powerhouse of the lab. This is a picture that our wedding photographer got of lab meeting Saturday night. So Carla and Margaret are amazing technicians that keep us all in line, and I don't think anything that I've done would be possible without either of them. And I love this picture. This is Carla and Margaret bringing a bunch of styrofoam to one of the styrofoam recycling events, and still smiling about it, even though it's not really a fun task to be doing. So Margaret taught me everything about doing cell culture, and I just recently learned she also taught me to do a Western blot without knowing how to do a Western blot. That was in my rotation. But Margaret takes on a lot of roles in the lab. And I don't know how she does it all. So she helps each and every one of us every day. She helps us, especially with tissue culture. She is the tissue culture fairy, so like, don't forget that when you leave stuff out. She's the one, you know, keeping us all in line. So she doesn't hesitate to jump on a project or help you out if you need it. And so Margaret's been helping test the the tin 2 isoforms and cell lines, so she's been helping me grow those cells, and she did the immunofluorescence. It's been a great experience. And Margaret also happens to do a lot of things outside of lab and a lot of volunteering and outreach, which I think is great. She's also a great roommate in New York. So a fun fact about Margaret is she has played in this Baltimore Gamer Symphony Orchestra. I keep missing the events, but right there. I don't know, so they play like video game music. It's pretty cool. And then there's Carla, who is the mom of the lab, who keeps us full of M&Ms. And she helped me so much learning how to do these telomerase assays and work with radiation and everything. So no one in our lab was doing those when I started it. So she helped dig through old lab notebooks and piece together protocols and helped me figure out what I was doing. And I can't thank her enough. She's visiting her daughter right now in Texas, so hopefully she'll watch this later and hear how much I appreciate all of her help. And of course, the rest of the lab, it's been a blast. A lot of good times with my lab mates, birthday parties, and miss Steve. I wish he would come back. Stella too. Some basketball rivalries, but we had a really good time. So once you eventually get rid of me, I'll miss you. I also thank Becca, who's my lab mate, my bay mate buddy. I think sometimes Becca and I spend more waking hours together than with anyone else. We've also been known as the wedding bay. So we both sat in our lab bay and planned both of our weddings. So it was, fun. And we spent a lot of time troubleshooting each other's experiments and encouraging each other that you can do the thing. And I don't think you're ever going to get rid of us because we're determined that we're just taking over the lab across the hall, and that's going to be the Alex and Bex way. But this is a picture from a few years ago when we had this big 6th floor group, third grader among us in Holland Labs. And Holland Lab will forgive you for moving upstairs, but that was a really great environment. A lot of great feedback and happy hours and science discussions with the Armanios and Holland Labs. So I think the CMM graduate program and showed the obligatory CMM anatomy photo. Been a really fun family with lots of Friendsgivings and food and parties and wineries and more food and drinking events. But especially thank Nina, who was my roommate for 2 years. Really good friend. We helped get each other through classes and oral exams. Iris, who kept us well fed by coordinating all of our dining experiences and making sure we all showed up. And David for taking most of these pictures and making us look really good. You can tell which picture he didn't take, I'm pretty sure. So I also have to thank Life, which is not just the word life, but Carol mentioned that I'm very into environmental sustainability. I spent a lot of my outside of lab and inside of lab time with this student group. And we would do things like scoop trash out of the bay, but also try to encourage people to be more sustainable and bring your own cup when you're getting coffee and ride your bike to work and stuff. Plant things, collect styrofoam. This is a regular just round table. So we collect that 70 pounds of styrofoam twice a year. And most importantly, we would plan sustainability happy hours where we can bring kegs on campus. Hopefully, encourage you to be a little more sustainable. So Carol also mentioned footy in the introduction. When people ask me why I play Australian football, I often tell them it's because you get to hit people. That's very therapeutic. But there's a lot more reasons than that. So you have really good friends outside of lab and in labs and around the country. You can sometimes take random trips to Iceland in March to play against some women from London. And I'd like to share one thing that I've learned. For a few years I played with the national team, and one of their sort of rules is to always be thinking of what to do next. So it's important on the field, like you get the ball and you give it to someone, but you're not done. And that's a good crossover to science. So once you get a result, there's like 8,000 things to do next, which is exciting, but it's also, you need to tell yourself that, because when something doesn't work, there's still something you have to do next. So that translates well. It also helps when your best friend gets a postdoc in Boston and you get to see her just about every month because you play sports together, and even if that means she's going to throw you on the ground, it's worth it. So there's lots of other friends and family that have been really helpful in this process. So I have some close friends from home, some ridiculous people, my Oswald team that usually wins when Pike and I are out of town. 2 best friends from college with mustaches everywhere. Dress-up parties, Kaylin again, not tackling me. And some of our watering holes, 2, basically one of which still exists. This guy, my husband Pike, has been very encouraging and helpful throughout all of this. Basically doesn't let me mope and whine about when things don't work. Just like, I'm gonna go do something else, or don't take yourself too seriously. He always looks pretty good. This was him at his thesis seminar, where his first question wasn't even a question. It was, "You look like you should be on the cover of Gentleman's Quarterly." I don't know, but I met Pike here, and it's been a fun number of years. He helped me make a dress out of Biohazard bags once. So, reminds me not to take myself too seriously. Then there's my family. My parents came here from Memphis. They've always been supportive of me, even when I tell them things like, when I was a kid, I said I wanted to be artist on the moon. Thought that was a little weird. I don't know if this is better career path. But always encouraging me to do whatever I want, that there's no limits and all of that. Also got a second set of parents now. My in-laws, they came here from Michigan, and Pike came with also sister and some cute nephews. And they've been very supportive of me as I keep saying I'll graduate someday, and I'm going to come on vacation but read papers in the corner. And my third set of parents, my godparents, have also been very supportive of me. They've taken me on trips and travel the world with them. It takes a village. My aunts and my yaya, lots of other people in my life have helped me get here and showed me that there's no limits to what you can do. And all of you know Yaya, who has sent us pounds and pounds of cookies over the years. This is her baking some cookies. So she's probably got the longest telomeres of us all. And she's been great support even when she asked me when I'm going to become a real person. And I'm going to end with something that we used to all do at the end of group meetings, but Carol got us to stop. We used to end all of our presentations with cute animal pictures. But Carol felt that inhibited scientific discussion because we were just talking about the animals. But I think I've already stopped this one. So this is Rita, who I refer to as my sister. It's my spirit animal, the sloth. That's Dustin's dog, who helped me make figures a few weeks ago while Dustin was out of town. Okay, that's all I got. Thank you.
So if anybody has any cute animal pictures, we're going to be showing them up in the 6th floor in 601 and have a little celebration of Alex. So you're all welcome.